Hi everyone, my name is Maddie and today I'll be presenting my summer research project on investigating sediment binding of wastewater pharmaceuticals, carbamazepine and ethanol estradiol in the Alawai Canal. Firstly, I'd like to thank Papa Jack for providing me with this wonderful opportunity to conduct a research project this summer. I learned so much and it will help me with my continuation project as I go along through the school year. I'd also like to thank my mentors, Dr. Dulai, Dr. Chan, and Dr. Trista McKenzie for being the best Alawai Canal sampling group, for providing Jaron, Tyler, and I with a workspace to analyze our samples, and for overall supporting me through every step of this process. Okay, so I'll just introduce my topic first because actually my summer project is based off of a continuation of what I did last school year in my honors independent research course. And so originally I was trying to determine whether there's evidence of wastewater from cesspools in stream water. And so we tested for concentrations in Manoa and Pololo stream along the Manoa-Pololo drainage canal, which runs right along the Lower School Autoline way. And we actually found that wastewater is actually present from cesspools, and they're detected at levels potentially harmful to organisms. So that led us to come to the question, what is causing these concentrations to persist, which led me to applying for the K Fellowship. Okay, so here in the Alawai watershed, we are actually surrounded by cesspools, and this is a map of like the Alawai watershed outlined in red, and here you can see Manoa stream, Pololo stream, and then red, the orange marks where I sampled during the school year, and then the green marks where I sampled over the summer, and the yellow dots represent individual cesspools in households. And so why should we be concerned with cesspools and cesspool wastewater in our streams? Oahu has nearly 11,300 cesspools which discharge about 7.5 million gallons of raw sewage into the ground each day. And for those of you unfamiliar with cesspools, they are these underground containers that store untreated wastewater, typically used by people who are not connected to the municipal wastewater system. So this means that when people flush their toilets, wash their hands, take showers, this wastewater gets stored into a container like the one depicted in the middle. Sludge piles at the bottom, while untreated wastewater dispels directly into the surrounding soil, which introduces contaminants including anticonvulsants, antibiotics, estrogen hormones, and beta blockers directly into the environment. And so again, on the far side, we have a picture of the Alawai watershed. And then I marked the sampling site at the bottom, and then Manoa and Pololo streams on the top side. Okay, so one way that we can detect whether wastewater from cesspools is present in stream water is through using wastewater tracers to detect their presence. And so the two tracers that we chose were carbamazepine and ethanol estradiol, which I labeled Tracer 1 and Tracer 2. So Tracer 1 is an anti-epilepsy drug used to prevent and control seizures. And then Tracer 2 is basically a birth control pill used by females. And so what we found over the school year is that concentrations of both were detected in water coming from Manoa and Pololo streams from cesspools. And so here I have the concentrations. There's about 6 pieces of anti-epilepsy pills per day, and then about 17 pieces of contraceptive pills per day for the day that I sampled. And so after we compared those levels to to previous studies that have tested the effects of pharmaceutical compounds on organisms, and so my concentrations are listed in yellow, and then the previous studies are listed in the white outlines. So Tyler and Jaron will be explaining more about the harmful effects on organisms because that's what they studied. They studied the effects of ethanol estradiol in stream water on tilapia. And so this is basically what I had from the school year. These are my results. As you can see, Stream water, which is less saline, flows at the surface. Alawai Canal water flows at depth because it's more saline. And so you have this sort of water gradient due to differences in salinity. And so that's how we sampled. We sampled surface and depth to get these concentrations. So I just averaged all of the results to give you a better picture of it. And as you can see, concentrations from depth were a lot greater than concentrations at the surface. So we know that there are traces of wastewater in cesspool wastewater in the pink and Alawai Canal water in the green. Okay, so after analyzing the data, we realized that concentrations in the green level were much higher than the pink level. So that led us to the question of, is desorption from sediments responsible for this? So desorption is basically after sediment particles absorb all of the concentrations, after they're bound together, it eventually releases it. So that's where I have the yellow dots being released from the sediment particles. And the other question is, are ground or suspended sediments a carrier for wastewater compounds? Okay, so the hypothesis is, if desorption is responsible for higher pollutant levels in the salty bottom layer, the green layer from the Alawai Canal, then I will see high levels in sediments and lower level water, deep water. And so these are the methods. This is my methodology. Okay, so initially what we did was we sampled along the entire length of the canal. So we started from the library end and went all the way to the harbor end of the canal. And in total, there were 11 different sites, so 11 green. And then we took 39 samples in total. And so the types of samples that we took were water samples and sediment samples. So basically, water samples consisted of surface samples and then deep samples. So surface, that's top level, and then deep level is the lowest that you can go, and then you just filter water through a pump. And then dredged sediment and then suspended sediment were the two types of sediment samples that we took. So suspended sediment was also taken at surface level, and then dredged sediment, to do that, we physically used a sediment catcher that we lowered into the canal, and then we pulled it up to get sediment. And this is what we did for analysis. So on the far side, those are my sediment samples that we had bagged up, and then we had to dilute the samples in order to separate the concentrations from the actual sediment particulates. So in the front, we have the suspended sediment samples, and the back is the ground sediment particles, and then we filter them to just extract the concentration. So that was about 1 milliliter of liquid. And then these are the water samples. So we plated both sediment and water samples into the ELISA kit to determine how much carbamazepine there was and how much ethanol estradiol there was at each of the little sites that we sampled at. And these are the results. So first I'm going to go over salinity and how that plays into the concentration levels detected in the canal. So again, we have sites A through K, all of the sites that we sampled from. And then on the top of the salinity chart, I labeled A through K. So the way that the Alawai Canal works is that there's actually a huge berm in the middle, which causes two larger, canyons. And so at depth, the red area, you have a lot of high salinity water. And then in the surface level, where we took suspended sediment and we took surface water, you have lower salinity. And then dredged sediment, that's essentially the entire black area as you go along the canal. Okay, so for tracer 1, carbamazepine concentrations. So the way that this works again is A through K along the y-axis, and then dissolved carbamazepine is for water, and then particulate-bound carbamazepine on the other y-axis is for sediment. And I guess each color represents a different type of sample that we took. So we have surface in blue, dredged sediment in gray, red is deep water, and yellow is suspended sediment. And so I guess to point out the most important part of this, you can see that from like A through D in gray, there's sort of like a decline in concentrations, which could suggest that because there's like that large berm in the canal, there's like a higher residence time for those concentrations because they can't like overcome the berm. And so for EE2, I still have to regraph my data because I want it in the format of the other graph, but basically these are the concentrations detected. There was nothing at surface level. Like we had 0 for the entire canal. For deep water, concentrations were not uniform either, and for suspended sediment, we had like a high level detected at site E, which is the exit area of the Manoa-Palolo Drainage Canal. And then for the sediment level, you can also see that sediment actually does hold some components of ethanol estradiol. And so these are the average concentrations from water and sediment samples that we took. I still need to add error bars, but basically these are the levels. So as you can see, it does follow the predicted that depth water does have higher concentrations of the pollutants. So blue is carbamazepine tracer 1, and red is ethanol estradiol tracer 2. And so blue, as you can see, at concentration 0.15 is much higher there at depth than anywhere else. And so in conclusion, so 1, wastewater tracers, carbamazepine and ethanol estradiol bound to sediment particles contribute to pollution. So because we detected concentrations of carbamazepine and ethanol estradiol in the sediment, we do know that sediment particles absorb wastewater tracers, which are later released through desorption. And then 2, salty deep water contains greater levels of pollutants due to longer residence time, and this may be because of desorption from sediments. So like I was talking about earlier, there's like that huge berm in the middle, the black berm. And if you look at the red triangle shape, that's where there might be a higher residence time because concentrations are much higher over there. And 3, suspended sediment particles are low in pollutants, which means that particles of sediment that just float in the water at the surface level don't really contain levels of carbamazepine and ethanol estradiol. Okay, so I just really want to say thank you to Papa Jack for supporting this fellowship, and then I want to thank again Dr. Dulai, Dr. Chan, and Dr. Mackenzie for helping me with my project this summer.